Okay, hope everybody had a good lunch. Let's resume if the next discussants are here. Dr. Landolf, are you ready to go?
Yes, sir, I am, Dr. Loomis.
Okay, let's then proceed with discussion of cancer studies in animals. It's your microphone.
Okay, you want my picture too?
Yes, please.
It says you cannot stop your video because the host has stopped it.
Ah,
Nope, still the same.
okay, well, why don't you just go ahead and speak and someone will probably get your camera turned on in a moment.
Okay, thank you. So first, I'd like to thank the staff, as many people have done before, for the enormous amount of work that went into compiling this 500, 600 page document. And I think they did a pretty good job on the animal carcinogenicity studies too. So I read over them. I want to go through them and just tell a court reporter I'll be on page 27 in Roman numerals. And so you can probably just copy a lot of this down from the document itself. So they said, the staff, that carcinogenicity studies of BPA have been conducted in male and female Fisher 344 rats, female Sprague Dawley rats, male and female Sprague Dawley in parentheses NCTR close parentheses rats, female Worcester first rats, birth rats, comma, male and female B6C3F1 mice, comma, male and female agouti C57 black 6J C3H/HEJ mice, comma, female CD1 mice and male gerbils. Statistically significant tumor findings are the following. So I first adopted the hypothesis that this was not a carcinogen, and let's see if that's true or not. So the staff found that from the literature, which they did a very extensive literature search, that in the alimentary system, there were hepatocellular tumors in male Sprague Dawley NCTR rats and female agouti C57 black, etc. mice. So that's positive. In the endocrine system, they found pituitary tumors in female Fisher 344 rats and male B6C3F1 mice, thyroid C-cell tumors in male Sprague Dawley NCTR rats. Then in the mammary gland, they found in the literature fibroadenomas in male Fisher 344 rats, adenocarcinoma and adenoma combined in female Sprague Dawley NCTR rats. And these, they say, were all statistically significant tumor findings. Then in the reproductive systems of the females, they found clitoral gland tumors and uterine stromal polyps. The stromal polyps are not malignant. And they found reproductive systems of males, testicular interstitial in parentheses Leydig, LEDYDIG cell tumors in male Fisher 344 rats. They also found lymphohematopoietic system in that system, leukemia in male Fisher 344 rats, lymphoma in male Sprague Dawley NCTR rats, and male B6C3F1 mice. In addition, the staff noted that multiple types of rare tumors were observed in several studies in male and female Sprague Dawley NCTR rats. I looked through that data. Most of them were ones here and there scattered throughout. And then they said there's more data on the animal tumor findings, which they listed below. In the male Fisher 344 rats, which was a 103-week feeding study, and BPA treated male Fisher 344 rats, the incidence of mammary gland fibroadenoma was significantly increased in a high dose, 2,000 parts per million, close parentheses group by pairwise comparison with control, with a significant dose-related trend. Great. In a 103-week feeding study in BPA treated male Fisher 344 rats, the incidence of testicular interstitial in parentheses Leydig, LEYDIG, close parentheses, cell tumors was significantly increased in both dosing groups. 1,000 and 2,000 parts per million, close parentheses, by pairwise comparison with controls with a significant dose-related trend. In 103-week feeding studies in BPA-treated male Fisher 344 rats, the incidence of leukemia was significantly increased in the high-dose, open parentheses, 2,000 parts per million, close parentheses, group by pairwise comparison with controls with a significant dose-related trend, NTP 1982. Then they discussed the tumors in the female rats, and they noted that in a 12-week oral study in BPA-treated female Fisher 344 rats, the incidence of pituitary tumors, likely adenomas of the adenohypophysis, was significantly increased in the low-dose group at 50 milligrams per kilogram per day. Tumors in the male Sprague-Dawley, open parentheses, NCTR, close parentheses, rats. In a 2-year continuous dose study in male Sprague-Dawley NCTR rats exposed to BPA in vitro and from PND1 until study termination, this is the Clarity BPA core study number 8, the incidence of rare hepatocellular carcinomas was increased with a significant dose response trend. So I starred that for emphasis. In a 2-year stop dose study in male Sprague-Dawley NCTR rats exposed to BPA in utero and in gavage from postnatal day 1 to postnatal day 2, the Clarity BPA core study number 4, the incidence of thyroid C-cell adenomas when it increased with a significant dose-related trend. I note that that's not a malignant tumor, but it's increased with a dose-related trend. In a 2-year stop dose study in the male Sprague-Dawley NCTR rats exposed to BPA in vitro and via gavage from postnatal day 1 to postnatal day 2, Clarity BPA study number 4, the incidence of malignant lymphoma of the prostate, dorsal/lateral lobes, was significantly increased in the high-dose group, 25,000 micrograms per kilogram per day, by pairwise comparison with controls with a significant dose-related trend. The incidence of malignant lymphoma from all sites was increased with a significant dose-related trend, NTP 2018. In the 1 and 2 year studies in male Sprague-Dawley NCTR rats exposed to BPA in utero and after birth for different lengths of time, arms 2, 4, 6, and 8 in Clarity BPA core study, close parentheses, multiple types of rare tumors were observed in multiple organs and treated groups of each of the study arms, except for arm 6, where only one rare type tumor was observed with none of the concurrent controls. Then they discussed the tumors in the Sprague-Dawley NCTR rats on page 29. And they said in the 2-year stop dose study in female Sprague-Dawley NCTR rats exposed to BPA in utero and via gavage from postnatal day 1 to postnatal day 2, which is Clarity BPA core study 3, the incidence of adenocarcinoma of the mammary gland and the incidence of adenocarcinoma and adenoma combined was each significantly increased in the 2.5 microgram per kilogram per study group. And they referenced NTP 2018. Next, they said in the 1-year continuous dose study in female Sprague-Dawley NCTR rats exposed to BPA in utero and from postnatal day 1 until study termination, which in parentheses was Clarity-BPA core study number 5, close parentheses, the incidence of uterine stromal polyps was increased with a significant dose-related trend, NTP 2018. Then they said in the 2-year continuous dose study in female Sprague-Dawley NCTR rats exposed to BPA in utero and from postnatal day 1 until study termination, open parentheses, Clarity-BPA core study number 7, the incidence of clitoral gland adenoma and adenoma and carcinoma combined was each increased with a significant dose-related trend. That's important. Next, they said in the 1 and 2 year studies in male Sprague-Dawley NCTR rats exposed to BPA in utero and after birth for different lengths of time, so open parentheses, arms 1, 3, 5, and 7 in Clarity BPA core study, close parentheses, multiple types of rare tumors were observed in multiple organs and treated groups in each of the study arms, NTP 2018. The tumors in the B6C3F1 mice, they said regarding these in 103-week feeding studies in BPA treated male B6C3F1 mice, the incidence of chromophobe carcinoma of the pituitary gland was increased in the high dose group, 5,000 parts per million, with a significant dose-related trend, NTP 1982. Then they said, quote, in a 103-week feeding study in BPA-treated male B6C3F1 mice, the incidence of malignant lymphoma and lymphocytic leukemia combined was significantly increased in low dose, open parentheses, 1,000 parts per million, close parentheses, by pair comparison with controls, NTP 1982. Then they talked about tumors in the female agouti mice. In the 10-month studies in these mice exposed to BPA in utero and veal lactation, then in feed from post-weaning until study termination, the incidence of hepatocellular adenoma and carcinoma combined was significantly increased in the high-dose group, open parentheses, 50 parts per million, close parentheses, with a significant dose-related trend. In parentheses, Winehouse et al. 2014, close parentheses. And I'm going to skip the rest of that because it's important, but it's not as important as those primary studies. So I was going to say, for me personally, as one of the members of the committee, I took that to indicate that there is a significant amount of animal tumor data that is positive and where the trend is dose dependent. And it's in males and females, and it's in rats and mice of different species. So therefore, for me, unless I hear something different, that convinced me that the animal studies are positive in rats and mice of different species. So although no study is perfect, nevertheless, the weight of those positive studies convinced me that BPA is indeed an animal carcinogen, in particular rodent carcinogen. And I can stop there.
Okay, thank you, Dr. Landolf. Let's go on with the discussion and turn to Dr. Bush.
Yeah, thank you, Dr. Loomis. And thank you, Dr. Landolf, for prepping the discussion here. I want to thank the OES staff for their Herculean effort, for delving into all of this data and creating this HID. So hands off to the teamwork there. And I have read the public comments from all 4 submissions. And I must say that some do raise compelling questions and statements. And so I'm going to start with Table 7. Dr. Landolf did a great job setting the stage here. My take on this is a little bit more pessimistic, I suppose, and let me explain why. So Table 7, which is page 54, 8 studies, and really the only studies of value that I see there are from the 1982 NTP results with rats and mice. The other 4 studies that were there had too few animals, and I question whether there is sufficient statistical power there. So delving into those 4 reliable studies on Table 7, really we're seeing the marginal increase in leukemias in males, p-value of 0.02. 26% of the controls actually have this as well. And we only see this trend in the high dose. Fibroadenomas were probably a little more convincing, but there's only 4 of 34 animals found with this kind of tumor. And the Leydig tumors, are common to the F344 strain. And so almost 75% of the controls got these tumors as well. So there's some data there, but I think it's marginal. And then when it comes to the rare pituitary carcinoma in the B6 mice, again, there was marginal increase when I'm looking at those trends. So we've got this data set, and then along comes the Clarity study, okay? When I first got wind of this a number of years ago, I was excited. This was going to be a well-designed long-term cancer study. We're going to learn lots of cool stuff and finally, be definitive on BPA. We're going to show that it's the smoking gun once and for all. And then, what happened? If we look at these different arms, looking at arms 1 through 6, many different endpoints, but from a macro view, looks like mixed results to me. There's some suggestive data, but I don't see it as being clear. There's some common themes like hyperplasia. Presumably that's some atypical hyperplasia, but nothing is clear dose response in my mind. And it's okay to say, well, that's arms 1 to 6. But arms 7 and 8, here we have the penultimate 2-year chronic rodent cancer bioassay. This now is going to be definitive. And what happened? There are some common biological phenomena, but again, it's not clear. I'd also mention that some of the public comments indicate that the statistical approach used in some of the analysis, it raised questions for me as well. So I do appreciate the description at the beginning by OHIA staff and the biostatistician discussing the methodologies there. So we have to balance this with the conclusions of the NTP itself, so the Camacho 2019 paper, where they determined that there's a possible relationship between the increased incidences of lesions in the female reproductive tract and male pituitary and exposure to the 25,000, the highest dose level. That's not very convincing of their own study, okay? Alluding back to some of the comments that Dr. Eastman mentioned previously. So is there a problem with this study? I do thank OHIA for identifying the shortcomings of the Clarity study, as others have described in the literature. There certainly are some limitations that were brought up in the initial presentation. I do share some of those concerns, but this still represents our best, most robust study. And I still believe that the results are mixed, and there still leaves some outstanding questions. So then what else do we have left in the animal studies? Well, we've got these, there are the other rat studies from table 26. These are, I think, for the most part, are of little value due to the small numbers, the short exposure times, and the lack of lifetime study duration. Now, that's not to say that there isn't something here, particularly when we consider that common theme of hyperplasia. So maybe there's something there. A few more notes here. Moving into the mice studies, similar problems, low numbers with 5 studies of any utility, and none of them went beyond 1 year. Again, hyperplasia is coming up, particularly in the endometrium and some possible hepatic effects, but the numbers are too small, and we're only seeing this in the high-dosed animals. The transgenic studies, various endpoints there, suggestive at best, and sporadic with no clear dose-response relationship that I can see. That common theme, again, emerging of hyperplasia. I didn't give really any weight to the xenograph studies because of the established cancer cell lines, which I always feel have limited value. And then the final group of studies looking at BPA before or after carcinogen treatment. There are various endpoints that one can pick, but nothing common. And so I see those results as mixed as well. And on page 101, OEA states, again, the limitations are small numbers, short BPA exposure, and only one organ tissue in many of these studies. So, where's the utility there? There isn't a lot. So, cherry picking the data a little bit, I think, can be problematic from a false positive perspective. So that would be my summary of the animal studies, and I will yield my time to the other committee members.
Okay, thank you, Dr. Bush. So third discussant, Dr. Lamero, what would you like to add to what we've already heard?
Sure. So thank you all for your great summary over at OEHA, and my the colleagues, your prior summaries have been very helpful, and I'll try not to be redundant in my comments. I kind of organized my thoughts more around the outcome rather than the individual study. So with respect to the individual outcomes, I think I just want to point out a part of the trends in cancer biology that are, I think, quite important, and that is, many decades ago, President Nixon declared a war on cancer, and in response, NCI put a lot of effort into trying to, better understand and treat cancers. And after quite a bit of time, they found that their methods weren't really quite working. And so they came up with the mouse models of human cancer consortium in the late 90s to address the fact that, rodent tumors are not that are spontaneous are not really the same as human tumors, and that if we want to deal with malignancies of humans, we need to have better models. And so I think, that field has really moved forward in thinking about having rodent tumors better recapitulate the pathologies that you see in humans. And there's certainly been a lot more success in that effort, as opposed to, for example, the NTP studies where we're still looking for spontaneous tumors. And I think that across the board, what I will say is I did not perceive, apart from the presentation of some rare pathologies, which I'll get to, that BPA modified the presentation of the type of pathology, and that each model, whether or not it's a spontaneous rat or mouse, or a transgenic, or the use of a carcinogen, or the use of a xenograft, basically produces the pathology of a tumor at that site that you would expect based on that model. And I don't see evidence of bisphenol A modifying it so much. The evidence that I think presented in general, apart for some rare things, was really changes in incidence, changes in latency, changes in growth, and changes in metastasis. So that's a kind of general impression I wanted to share with you all and kind of put it into context of how I think about modeling cancer. So there was several studies that reported lymphomas. We have the NTP 1982 study where we saw the male B6C3F1 mice had an increase in malignant lymphoma among the lower dose they tested. So they had 1,000 ppm of BPA in that study, and saw 8 of 47 of those male mice with this malignant lymphoma. Only 2 of 47 had it in the controls, and that was at 3 of 45 in the highest dose group at 5,000 parts per million. The next and only other study in whole rodents that looked at that observed lymphomas was the Clarity study that we heard about. And let's see here. We have the stop dose 2-year study. So just looking at the early window of susceptibility, saw an increase in the 25 mg per day group, which was the highest dose group. And they didn't see them anywhere else, but there were 4 in that high dose group. And so that individual group was significant statistically and also contributed to a significant trend of this malignant lymphoma with a p-value of less than 0.01. And this is notably an uncommon neoplasm in Sprague-Dawley rats, and the controls were within historical ranges, is how they defined less than 1% of the historical range was part of the definition that we had provided to us. And then in addition to that, and the Clarity, there was the observation of a different kind of rare tumor, which is called a histiocytic sarcoma. And these are often considered to be related to lymphomas. So I wanted to point that out. And that was also in the same dose group. With respect to leukemia, another hematopoietic cancer type, we saw in the 1982 NTP study, the males had increased leukemia in the high dose group. There were 23 out of 50 of the male F344 rats that had 2,000 parts per million of BPA in their food had leukemia, whereas only 13 of 50 of the controls had it. So it practically doubled. And that was also a significant trend with a p-value of 0.02. Again, the controls were in the historical range. This wasn't observed in the females, although you could say that there was a trend where each group had 50 females and the controls had 7 leukemia, 7 female rats with leukemia. The middle group, it went up to 13, and then the high dose group, it went up to 12. So in the presence of BPA in females, it kind of doubled, but it was not significant. Then further evidence for leukemia also came, sorry, I'm just skimming here. I just want to point out in the NTP 1982 study of mice, there was no evidence of the leukemias being significant, but there was a bit of a trend in terms of there was none in the control group, and there was 1 in the middle dose group and 2 in the higher dose group. So, rare events are hard to statistically capture. So it's a bit underpowered perhaps. And then in the Clarity study, it's done many years later in 2018. So you're getting a little more fine understanding of diagnosing or calling the leukemia. So they called it granulocytic leukemia rather than not otherwise specified, which is what we found in 1982 NTP. The male Sprague-Dawleys or the NCTRs of that Clarity, we saw each dose group had one of these granulocytic leukemias, but not in the controls. And again, that's a rare tumor in those as well. So we're seeing that across 2 species, 2 studies. They're getting increased leukemia. There's a lot of studies related to mammary gland pathology and trends in terms of onset and growth and metastasis. And I think it really, as I, was talking at the beginning, is a great example of this kind of, you get what you would expect with the model. So when you have a spontaneous rat mammary tumor, you don't tend to get much more beyond a hyperplasia. It's not, they don't tend to make malignancies, and they don't mimic the pathology really of human breast cancers all that well. But we do see hyperplasia and let's see here, the Clarity study in the female 2-year that I think was already brought up. It was also a similar thing that came up when someone used a pump to deliver bisphenol A osmotically. They also later saw adenocarcinomas in the pump model. They changed the dosing around a little bit with the pump model, expanded the window from in utero to lactation, and again, basically saw hyperplasias and adenocarcinomas at a later time point, which was only postnatal day 140, so 6 months. The average length of their life is several years. So, the scope in terms of, I know it was brought up about the sample sizes. I mean, I think low sample sizes are really going to bias your results towards the null, right? Because you have less power to detect effect. So similarly, if you're not looking for as long, you have less opportunity to observe an overt malignancy. We also had the Wister-Firth rat model, so a different strain or substrain of the rats, and they too had hyperplasias and also described some DCIS. And those female rats, ductal carcinoma in situ. So basically it looks like a cancer, but hasn't done the invasion part. So it's a kind of pre-neoplasm, we could say. Then in mice, there's a couple studies. Obviously there's a limitation with the relevance of the osmotic pump as a route of exposure, but these folks used gavage and a gestation period that was kind of similar to the rat study. They observed lipoma in the mammary gland, and through 3 and 8 months of age weren't observing any mammary gland tumors, but by 14 months there was some of this hyperplasia that I brought up before. And then when you move into a transgenic model that mimics the HER2 positive human breast cancer, and that one's called MMTV or ERB2 for short, it has a much longer name. They use drinking water, BPA delivery, and this model is kind of the gold standard and research on HER2 positive human breast cancer. It gets mammary adenocarcinomas, and they're capable of metastasis. And in that study, they were able to observe increased tumor multiplicity and tumor volume, which is kind of a surrogate for growth, in addition to significantly increased lung metastasis. And that was at both doses that were used in that study by Jenkins 2011. And then there was another study that used the same mouse model, but instead used subcutaneous dose. So not as gold standard in terms of mimicking the human condition, but they too saw a reduced latency, and it went from 37.5 weeks or so in the control group to 35 weeks in the 50 nanogram per kilogram group, and down further to 32 weeks in the 500 nanogram per kilogram group. And those were significant. And then they saw increases in some non-lesion items, but that are consistent with kind of more mechanistic understandings of things like, but I'll just mention them here since we're talking about that study, and that was increase in terminal amphiboles, which are full of pluripotent, rapidly proliferating cells during during puberty, and they also measured increased proliferation of the epithelium from those mice exposed to BPA. There was another study in mice that looked at hyperplasia or saw increased hyperplasia and DCIS with bisphenol A. Then in clarity, we heard about that already, but basically there was one fibroadenoma, and there wasn't really much in the way of neoplastic lesions. So in that spontaneous rat model, not really anything going on at all. There was a couple different studies, maybe 5 or 6, that used a carcinogen, either DMBA or MNU, as a co-exposure with bisphenol A. Excuse me, and most of those studies, they saw an increase in the mammary tumor incidence. A handful of them saw an increase in tumor multiplicity. I don't think tumor multiplicity is as relevant to the human condition, because women don't usually present with multiple independent tumors when they get diagnosed. And this was, excuse me here. Both in female rats and in mice using these co-exposures, it was reported that there was decrease in mammary tumor latency, and a couple of the studies indicated that there was higher proliferation in the lesions. Several of the studies did not report the pathology. I thought the pathologies were kind of typical of what you see using DMBA and MNU. So for example, DMBA usually produces squamous cell carcinomas that are not really considered very similar to humans. So these studies are, you know, supportive secondary evidence, but I wouldn't hang my hat on them if it was the only information we had. And then there's a couple xenographs with BPA exposure before or afterwards, and they do see increased growth of these xenographs, which are human breast cancer cells implanted on mice, suggesting, you know, BPA can be a promoter. And then yeah, I think I'll just leave that as the end of the mammary. There's a couple of studies that addressed hepatocellular carcinoma. So in the clarity study, there was an increased incidence of that in the dose groups, but that were higher. So this is a rare carcinoma to find in rats. They saw 2 of 24 in the 250 microgram group, 1 of 24 in the 2,500 microgram group, and then in the 25,000 microgram group, there was 3 of 19 males NCTR rats that had this rare hepatocellular carcinoma, but none of these lesions were observed in the lower doses of that clarity or the control. So that was a significant trend at 0.01. And then in the agouti, C57 black silix spread to the C3H, HEJ, dietary BPA was used in the White House study that our first speaker talked about, where we saw hepatocellular carcinomas in all the BPA treated groups and none in the controls. And again, that was an significantly increasing trend. So we have 2 species where we have a significant trend of a rare tumor, and none in the control. So I think that was noteworthy. There's a fair bit of prostate tumor research that's gone on, and a number of studies through the years. Most of this comes out of Gail Prin's lab. I don't want to get into the details of every single one, but I will say that, let's see here, if I can. Sorry, just lots of notes here. There was one study that didn't come out of her lab in 2003, the Ichihara study, the F344 males had been evaluated up to 65 weeks old. They had no prostate tumors or pre-neoplastic lesions, and dose groups ranging from 50 to 120,000 micrograms per kilogram per day. The Prinz lab tends to use this Sprague-Dawley model instead. So they did not report any BPA related neoplastic or pre-neoplastic effects in any region of the prostate as part of Clarity. But OEHA reported that when they looked at the supplemental data and in a table of that peer-reviewed publication, and this is in table 23 of our workbook, there is a statistically significant increase in pre-neoplastic, high-grade prostatic intraepithelial neoplasms, which are referred to as PINs. And this was evidenced by a significant trend, but it was prostate region specific. And then Prinz like to use this a pump system where they provided testosterone and estrogen to more mimic the human condition of an aging human man. And in those cases, they observed that BPA increased microinvasive carcinomas and PIN. So there were several studies that among their group that found that using this hormone supplemented situation. Okay. Sorry, I'm just going down here. There was also a xenograph of a human prostate cancer cell line that's commonly used called LMCAP. And they had increased growth when exposed to BPA. And then if BPA was, if the rats were exposed to BPA during or after a human prostate xenograph, that also increased the grade of PIN in that testosterone estrogen model that Prinz uses. I believe I might have said that was a pump, and actually it's more correct to call it a pellet, so excuse me. NTP in 1982 observed the increased Leydig cell tumors that we heard about before, so I won't touch on that. There was a couple of reports of pituitary tumors across female rats and male mice. So the female F344s had increased incidence of pituitary adenomas, and the low dose group at 50 mg per kg in the NTP 1982 study. So that was at a p-value of 0.05. They saw none of these pituitary adenomas in the control group. In the 50 mg group, they saw 4 out of 10 of these rats had the pituitary tumors. Only 1 of 10 of the rats at the 200 mg dose group got them, and then 3 out of 10 got them in the 400 group. So not a linear dose response, but an absence in the controls, but not the treated. And then with the male B6C3F1 mice that the NTP 1982 study used, they also saw an increased incidence in pituitary tumors. However, they were a different subtype called chromophobe carcinomas. These appeared in the high dose group, none in the controls, kind of an intermediate amount in a middle dose group, so just barely reaching it, an increased trend significantly with a p-value of 0.046. So a little bit of evidence of pituitary tumors increased in 2 species. Then there's a handful of uterine, reproductive, and gonadal type female lesions. I think most of these are not really some of them are not neoplasms or necessarily pre-neoplastic, but I'll just point out that in the Clarity study, there were 2 observed fibrosarcomas of the clitoral gland and the BPA 2.5 and 250 mg per kilogram dose groups among animals that didn't have those clitoral lesions that were described earlier. So there seemed to be, that's been a rare one. Then there was other study that looked at subcutaneous BPA being provided to CD1 mice, and there wasn't really any significant changes. So overall, I think that category of the gonadal region, we'll call it, I thought that in general is fairly unremarkable, but I did want to point out the fibrosarcomas. I last want to just highlight a few things about what I call miscellaneous rare tumors. I went through, you can find a summary of those on table 25, but I would like to point out that it's missing osteosarcoma and histiocytoma. So I went through each of the individual tables of Clarity that were provided related to rare tumors in particular, where I wanted to see if there were any that appeared in both sexes in order to make me feel more confident that they might not just be simply spurious results. And what I found was that we had, as noted in table 25, the males from the 1-year perinatal chronic, or where they start in the perinatal period and proceed through the rest of the study, they had small intestine carcinoma observed, and the females from the 2-year chronic continuous dose study that started in the perinatal period, they also had small intestine adenocarcinoma observed. And then when looking at just the female and males from the 2-year chronic study, they had the common histiocytic sarcoma, which can arise de novo, but is actually also known to arise from B-cell lymphomas. So again, tying back to the evidence that there might be something going on with rodent lymphoma risk related to BPA exposure. And then the 2-year stop dose in males had osteosarcoma, as did the female 2-year continuous dose study. So I thought that it was interesting that we had a couple of the rare tumors appear more than once across both sexes of the rats. And I think I will stop there. So thank you for listening to my overview.
Okay, thanks, Dr. Lamero. Let's see whether any of the other assigned discussants have anything to add that we haven't heard already. Okay, it seems there's nothing else. So we'll move on to the assigned discussion of the 10 key characteristics of carcinogens. And we'll We'll take these mostly in order, but Dr. McDonald has number 1 and number 5. So while you have the microphone, Dr. McDonald, if it's not too confusing, I'll just ask you to do both of those, and then we'll go back to the established order.
Sure, that would be fine. I also want to acknowledge the work done by AWIHA to synthesize the data on bisphenol A. Once again, you've managed a Herculean lift. The amount of studies and information on this chemical is daunting. And so thank you for pulling this all together. I also want to thank the public comments of US FDA, ACC, NRDC, and PIA. The case of bisphenol A is a difficult one. The epidemiology data are troublesome because of exposure assignment. The animal cancer data are suggestive, but the largest pivotal study, Clarity 7 and 8, where you have lifelong exposure and you really should see effects. Instead, the effects are unremarkable. Yet we have a rather large mountain of mechanistic data that shows bisphenol A causes a wide range of effects, including DNA damage, oxidative stress, altered hormone states, changes in cell function, immortalization, proliferation. So these mechanistic traits or key characteristics of a carcinogen are going to be discussed in the next 10 sections. And the first one is whether bisphenol A is electrophilic or can be metabolically activated. So a key characteristic is electrophilicity. That is whether the molecule itself, or more likely or more often, the metabolite of the chemical is reactive or electron seeking. That is whether the chemical or its metabolites bind to electron-rich cellular macromolecules like DNA, RNA, lipids, proteins, forming addition products, which we usually refer to as adducts. Binding to DNA is good evidence of electrophilic activity. So bisphenol A can be metabolized by the cytochrome P450 enzymes and peroxidases, which Dr. Ricker very nicely walked us through this morning. These pathways are quantitatively minor, less than 10%, but they still probably carry all of the concern. These pathways form a variety of electrophilic compounds. The most notable is the bisphenol A 3,4-quinone, often referred to as BPAC. There's a semiquinone intermediate, an arene epoxide intermediate, an isopropene phenyl radical, and then there's another electrophilic compound that then dimerizes to bisphenol A. And with all this redox cycling and quinone formation, you get a lot of reactive oxygen species formed as well. The DNA adduct studies have mostly focused on the quinone adducts. The primary ones are with guanine and adenine. So bisphenol A 3,4-quinone, or BPAC, has formed DNA adducts that have been identified as 3-hydroxy bisphenol A and 7-guanine, and 3-hydroxy bisphenol A and 7-adenine. So the DNA adducts have been measured following treatment with BPA in various systems, including human in vitro, animal in vivo and in vitro, and of course in cell-free systems. Importantly, DNA adducts have been measured in human cells in vitro at low concentrations. For example, DeFlora et al. In 2011 measured DNA adducts in both normal and prostatic tumor cells with fairly low concentrations of 200 nanomolar, and also down to 1 nanomolar when treating long-term. There's an increase that it was not quite significant, but close. There are also studies that have measured DNA adducts of bisphenol following oral and interperitoneal dosing of rats and mice. Unfortunately, these studies used very high doses, 200 mg per kg, and there are no other lower dose studies that were summarized. These studies found DNA adducts in the liver of rats, and both DNA adducts in the liver and mammary gland of mice, of CD1 mice. Interesting, the same adducts were found in liver and mammary gland epithelial cells. So DNA adducts are not a required step for carcinogenesis, but they certainly demonstrate that these metabolites are electrophilic and capable of DNA damage. N7 guanine adducts in particular are very good biomarkers of internal exposure of activated carcinogens. There are some researchers, though, that have questioned the biological significance, since they do not persist and are they form readily and not likely to be mutagenic. But usually when you have these adducts, they're accompanied by many others that are potentially mutagenic, as well as we have a lot of reactive oxygen species being generated along the way. So with respect to oxidized DNA, there's a significant evidence that bisphenol A causes oxidative DNA changes, which are believed to be due to the reactive oxygen formation, reactive oxygen species. So I'll discuss DNA oxidized bases in the next section in oxidative stress, which will now be very soon. But the reactive oxygen species and the DNA oxidation probably underlie a lot of the DNA strand breaks and other genotoxicity that you'll hear from the next discussant. Let me make one more point on this topic, is that bisphenol A, because it forms quinones, it loves to bind to cysteine residues on proteins. In a recent study, for example, Hu et al. In 2022 examined protein adducts in the liver of treatment of rats with a single high dose or repeated lower dose, and modified proteins included superoxide dismutase, catalase, glutathione transferase, and these, of course, are important because these are the key antioxidant protective enzymes that protect against the oxidative damage. So just in sum, bisphenol A is an electrophilic chemical with the potential to cause DNA and protein adducts, as well as reactive oxygen species. So that's that section. Okay, so shall I move on to oxidative stress then, Dr. Loomis?
Yeah, go ahead.
All right, so oxidative stress, key characteristic of carcinogen number 5. Another key characteristic of carcinogens is oxidative stress, and that, as you all know, is really the imbalance between reactive oxygen species, or ROS, or reactive nitrogen species, relative to antioxidant properties. So these reactive species may add to carcinogenicity through DNA alterations, changes in cell cycle control, but it really is the cell's ability to maintain the balance between oxidation and reduction that's important for cell development, growth, and survival. We, of course, during normal metabolism, generate reactive oxygen species, and we have many antioxidant molecules, enzymes, efficient and regulated pathways to scavenge these species and prevent toxicity. So really it's when you get the host behaviors or chemical exposures that tip that balance that leads to toxicity. So reactive oxygen species can be formed during the metabolism of bisphenol A, probably through the redox cycling of these quinones and semiquinone metabolites. They also could be formed through other oxidation reactions. So in the bisphenol A studies of oxidative stress, there are a number of biomarkers that are used, mostly for oxidative DNA damage. They assess 8-hydroxydeoxyguanosine. And as noted in that Stephenson 2020 review, this marker is easy to measure, but it does have the weakness of high inter and interday variability because of many confounding sources, such as food, exercise, smoking. Some other studies looked at lipid peroxidation or oxidative damage to lipids, and these were often measured using the markers of malondialdehyde or 8-isoprostane. And then other studies use general markers such as glutathione levels, glutathione dimers, as well as changes in the function or titer of protective enzymes such as glutathione transferase, glutathione peroxidase, superoxide dismutase, catalase, and others. I should step back just briefly. We really should take some caution when applying this key characteristic in particular, because oxidative stress is caused by many non-carcinogens as well. Any chemical that causes systemic toxicity or inflammation, can cause inflammation. And you often see changes in these same markers as well. So I think we should focus on oxidative stress as it relates to the potential mechanisms, especially at non-toxic doses, and the potential outcomes such as gene tox and receptor and cell signaling. So bisphenol A has induced reactive oxygen species in hundreds of studies, in humans and animal cells in vitro, including low concentrations at or near human expected levels in some populations. In animals in vivo, again, including quite low doses on the order of low microgram per kg, induced a ROS in many tissues and cell types. In human observational studies, we must control and consider the factors such as co-exposures, disease state, and smoking status. And also some of the human observational studies have the same limitation that we saw in the EPI studies where, some use single spot urine samples for exposure assignment. However, many others use multiple exposure time points. So that really strengthened their studies. There were 13 observational studies. 6 showed no or negative association, but the vast majority were positive, and these did include some studies that had repeated urine samples to improve exposure assignment. With respect to human in vitro, there's over 24 studies of bisphenol A in human cells that reported significant changes in a variety of cell types. These included lymphoblastic cells, breast cancer cells, colon cancer cell lines, neurons, kidney, blood cells, liver, and lung cells. Interestingly, some of these studies showed that ROS not only forms and can be measured increased in the cytosol, but also in the mitochondria. So bisphenol A metabolites do accumulate in the mitochondria, and they lead to energy shutdown, cell death, which of course then kicks up cytokines and can lead to inflammation. Animal in vitro, there were many animal in vitro studies that showed increase in ROS markers in a dose response fashion in the blood and in the tissues, including ovary, liver, brain, testis. Also increases in these markers, including malondialdehyde, were seen in animals given greater than 5 mg/kg. But there were a few studies that showed markers elevated even after low doses on the order of microgram per kg. Let me just give you one example that is one of the longer term studies in this group, Wang et al. In 2019. It was a 10-week drinking water study. It's a good example in mice. Longer term oral intake at 50 microgram per kg induced ROS markers in the serum, colon, and liver, as well as corresponding decreases in the activities of the enzymes that protect against oxidant status, such as superoxide dismutase and catalase. There were clear increases in inflammatory cytokines. There were clear reductions in mitochondrial function in the tissues, and there was a clear increase in caspase genes being expressed, as well as functional enzyme activity in the liver and colon, suggesting BPA-induced apoptosis. I thought that was interesting just because there's quite a number of studies that looked at in vitro activities of apoptosis in cancer cells, such as that reviewed by Namiri in 2019, where they showed BPA inhibited apoptosis in some studies, where actually inducing apoptosis in others. One of the key review papers that we highlighted was that of Amjad in 2020. It summarized numerous animal studies that had been conducted where bisphenol A induced oxidative stress that was alleviated by co-exposures to a variety of antioxidants like catalase, small molecules like vitamin A, C, and E, melatonin, lycopene, ginseng. Nearly all of the antioxidants reduce bisphenol A oxidative stress, lipid peroxidation, and DNA damage. I think it just really points to the complexity of studying bisphenol A effects in humans, and there's this constant, always the inner balance between that oxidation and reduction that's always going on. All right, I'm almost through here. I've just got animal in vitro. There were about 36 studies in rodent cells from a variety of tissues. Almost all found significant increase in ROS markers. For some cell types, high micromolar concentrations cause cell toxicity, cytotoxicity, but there were actually the majority of the studies in mammalian cells showed lower non-cytotoxic dose still resulted in ROS formation. So in sum, I think it is clear that bisphenol A causes oxidative stress. It causes oxidative stress at high doses, but also importantly, it does so at lower doses as well that are non-cytotoxic. Bisphenol A induces reactive oxygen species that appear to be involved in altering cell signaling pathways, promoting cell proliferation and survival, as well as DNA damage. That's it. Thank you.
Thanks, Dr. McDonald. Let me just point out that it is 10 minutes to 2, and remind the remaining discussants that you can assume that we've all looked at the report and the data to the extent we need to. So please just summarize your observations about the studies and the key takeaway messages that you think the committee should be aware of. We'll go on now to Dr. McDonald with key characteristics 2 and 3. I'm sorry, Dr. Eastman.
Thank you, and I'll try to move through this fairly quickly. So key characteristic 2 is really, does the agent exhibit genotoxicity? And there is evidence that bisphenol A is genotoxic at multiple endpoints in vitro, although the results are somewhat more mixed than seem presented in the document to me. For example, the document says it causes mutations, and so if you look at this, it's negative in many, the Ames test in salmonella bacteria, been tested many times, been negative in yeast, it was negative in 4 mutation assays in standard tests in animal cells, but it was positive in 2 non-standard tests in human cells. It was also positive in one dominant lethal study in rats and negative in one. So you get this sort of mixed picture, but there's enough evidence here that it does look like it is positive, certainly in vitro, and that's for chromosomal damage, DNA strand breaks, adducts as well, and oxidative damage to DNA, as indicated by Dr. McDonald. The evidence for genotoxicity in vivo is limited and is more problematic, and I'll give a couple examples. So about 20 years ago, there was a very high-profile study by Pat Hunt and colleagues that reported that bisphenol A caused aneuploidy and related effects in germ cell, and it's in the document. However, there was a group from Europe that actually had funding from the European Commission to work on that exact same type of study. I should say there are only about a handful of labs in the world that do these types of studies. So it was actually very fortuitous. This other group by Pacchiarati et al. Had funding to repeat the study by Hunt, and they were unable to repeat it. And so the repeat study is also in the document, but you don't see it in the context. The one was quite dramatic sort of result, but the follow-up result by a very reputable group of investigators was not able to repeat that work. And then there's some other issues that come up in vivo where you have one group in actually what I consider actually quite a poor study. It reports chromosomal damage at very low concentrations of bisphenol A. Similar results are not seen at much, much higher doses, 1,000 times higher doses by, result labs that are considered to be quite reputable. So you get this sort of issue. It's kind of a messy pattern, but certainly there's some evidence for genotoxicity. I'd like to put this, if I could, in a little bit broader context, since most of my career I was involved looking at genotoxicity related to phenolic compounds, and Tom McDonald was involved in some of this many years ago. But so the metabolism and toxicokinetic information to indicate to me that positive results, when they are seen, would likely be much more common or restricted to the high doses of bisphenol A. If you think about the toxicokinetics and metabolism that was presented, it's very similar to other phenolic compounds in that initially when the body is exposed or cells are exposed to these phenolic compounds, there's phase 2 conjugation takes place. So that basically you have conjugation with glucuronidation and sulfation occurs, and that really predominates. But as you get to higher concentrations, then you start seeing bioactivation by cytochrome P450 monooxygenases. And these will form reactive intermediates such as your quinones, but generally there's fairly substantial levels of reduced glutathione there, which will conjugate with this. So are planned to inactivate that. But as you go up to higher doses or higher concentrations, then you'll start seeing more covalent binding to macromolecules such as proteins and DNA. And that's sort of a common sort of classical toxicology perspective. And that's the way I look at this or interpret, and that seems to be consistent with the evidence. So this suggests to me that that there should be a sort of strong nonlinear component to the dose response curve. And indeed, the United Kingdom's Committee on Mutagenicity concluded that the dose responses of similar compounds, hydroquinone and phenol, are likely to exhibit a threshold response just because of these multiple protective mechanisms in vivo. Another point I'd like to point out is that this phase 2 metabolism, which tends to be quite efficient, is found for phenolic compounds both in the intestine and in the liver. So it becomes important when you evaluate studies to identify the route of exposure so that, is this relevant to sort of oral exposure, or is this a unique sort of thing? So we talk about osmotic pumps implanted. The compound is reaching systemic circulation without directly going through the intestine or the liver. So you might expect to see somewhat different results than if the compound was given orally. So in sort of summary, I think there is certainly evidence that, and I should say, Dr. McDonald went through the evidence for there's oxidative damage to DNA in multiple studies reporting this as well. So there is certainly evidence bisphenol A is genotoxic in vitro, but I would consider the evidence in vivo to be much more limited, certainly for traditional endpoints. And that's kind of my summary for key characteristic 2. Key characteristic 3, it alters DNA repair or cause genomic instability. As indicated in the report, overall, there were a relatively small number of studies that looked at the ability of bisphenol A to alter DNA repair or cause genomic instability. And a few of them reported the bisphenol A decreased capacity for cells to repair certain types of DNA damage in various cell types. And then a few others reported decreased expression of DNA repair enzymes. So I consider these evidence in this area to be fairly limited. So there's maybe some suggestion there, but it's not certainly convincing by any means that bisphenol A may cause some effects on DNA repair, but it's not, again, it's sort of in the limited evidence or suggestive evidence category. So that's really my comments for those 2 key characteristics. I'm happy to answer questions if people have them, or maybe we'll wait till later.
Yeah, let's move through all the key characteristics and then we'll open it up for questions. Okay, next up, Dr. Bisphenol, key characteristic 4.
Thank you. Let me get to my notes. Well, I want to also recognize the work of all contributors to this nearly 600-page report. I'm sure it was a major undertaking and lots of work went into that. The document is a good read and it reviews a large body of work. As we heard today, the literature on the cancer-causing potential of BPA is quite rich, but focusing on the epigenetic effects, the report identifies, if I read it correctly, 413 articles published in this topic. I myself did a quick PubMed search and found 128 review articles only on this topic. This is quite astonishing considering that investigating the epigenetic effects of this chemical only gained momentum about 16 years ago. So that was basically due to publication of 2 seminal studies, one by the Prince Group in 2006, and the other one by Journals Group in 2007. This latter study, I'd recommend inclusion of this second very important study, which was published in PNAS, somewhere appropriate in the report. Following the publications of these 2 studies, there has been a flurry of research into the epigenetic effects of BPA and other endocrine disrupting chemical. The report summarizes many of these studies, and the presentation given earlier today highlighted some of the findings of those studies. I'm not going to repeat what is written in the report or was presented today, but just to give a brief overview of my gathering from reading the literature on my own and reviewing the report. My take is that there is basically 3 distinct but often interrelated epigenetic modifications that are associated with BPA exposure. Among these, aberrant DNA methylation is the most studied one. Relatedly, studies on DNA hydroxymethylation are also beginning to emerge. These studies have been performed in vitro, in vivo, and in human populations. To much lesser extent, studies on histone codes and non-coding RNAs have been also performed in cell cultures treated with BPA or in tissues and organs of animals treated with this chemical. The histone modification studies have focused on a few active or repressive histone marks, occasionally measuring enzymes that catalyze these reactions, and sometimes quantifying the expression of the associated genes. Likewise, studies on long non-coding RNA, specifically microRNAs and long non-coding RNAs, and BPA exposure have mainly been in vitro and in vivo experiments, and fewer human studies are there in the literature. I'm not going to go through the histone modification or non-coding RNA studies, as many of these studies are extension of the original DNA methylation studies. And most of the comments that I will make for DNA methylation studies will also apply to those other studies. As for DNA methylation studies, a large number of in vitro and in vivo experiments and human studies have shown that exposure to BPA is associated with either gain or loss of DNA methylation in a single gene or multiple gene. The report refers to these studies as individual gene methylation studies. There have also been reports of association between BPA exposure and DNA methylation at a global level. These are referred to as global methylation studies in the report. What I would like to note is that with the exception of perhaps a couple of studies, the global studies, as they are referred to in the report, are not truly genome-wide studies, because they have either used methylation array, which interrogate only a small fraction of the CPGs of the epigenome, of the genome, at best less than 2%, or they use ELISA or immune precipitation or other enrichment-based method to analyze specific repeat elements as a proxy for the CPG content of the entire genome. So as it stands, there is a need to study the effects of BPA on the whole methylome, perhaps using more advanced techniques such as NGS-based platforms. Based on the in vitro and in vivo data, there is evidence that exposure to BPA is associated with aberrant DNA methylation, both in individual genes and in gene panels. These associations are mostly cell type dependent, as is shown in cell culture experiments. They have also been shown to be tissue specific or sex specific in some animal studies. A wide variety of cell types from different species have been treated in culture with BPA, at varying doses, mostly in the nanomolar to macromolar range. Following the treatment, some, but not all of these cell types show changes in methylation status. For example, in the promoter region of a gene or in repeat elements. Again, methylation changes are detectable in some, but not all doses. Also, establishing a dose response relationship has not been straightforward. Pathway analysis of the differentially methylated genes has been performed in several of these studies. The results show enrichment of molecular pathways that are implicated in cancer, neurodevelopment, and metabolism and reproduction, among others. Let me see. Several of these studies have also measured the expression of enzymes that catalyze DNA methylation. Both overexpression and underexpression of DNA methyltransferases, both the de novo and the maintenance DNMTs, have been observed in vitro and in vivo experiment. The relationship between enzymes level and the methylation status of the tested gene has not been direct as can be expected. And more or less similar findings have been reported from animal studies after in utero or lactational exposure to BPA. Again, I want to underscore the tissue specificity and sex dependency of the effects that have been reported in most of these animal studies. As for human studies, the focus has mostly been on mother-child pairs or adolescents and adults with environmental exposure to BPA. There are also studies in adults occupationally exposed to this chemical. Although associations between DNA methylation status and BPA exposure have been found in some of these studies, the results need to be interpreted quite cautiously. As we heard all day today, a main concern is the reliability and precision of exposure data for BPA in human studies. To make matter more complicated and complex, there is the issue of epigenome plasticity, which can significantly impact human study. As you all know, the epigenome can change by both physiologic and pathologic conditions, developmental stage, normal aging, exposure to a wide range of chemicals and agents, lifestyle factors, diseases. These all can affect the epigenome. So it's tremendously challenging to account for these factors in the epigenomic studies in human population. In the case of BPA, this situation might be even more complicated considering the pervasiveness of this chemical in the environment. Its complex pharmacokinetics, particularly in its short half-life and rapid excretion, and most importantly, lack of long-term exposure biomarkers for this chemical. I'll briefly mention some examples of the limitations of the published studies in humans. For instance, in mother-child pair studies, spot urine samples from mothers were collected for BPA measurement in order to find this association with DNA methylation in fetal tissues, placenta, cord blood, or peripheral blood from offspring 2 years up to 14 years after birth. Again, as we heard all day today, the accuracy and representativeness of a one-time measurement of BPA in mother's urine is at best questionable, especially when it's used to estimate the gestational exposure or the newborn's exposure years after birth. A further complication is the continuous exposure of these newborns to other sources of BPA and the constant alterations of their epigenome as they continue to grow. The same concerns also apply to studies in boys and girls and adults whose urine or serum samples were taken at a single time or twice a year in order to make an average for annual BPA exposure. Also, in many studies, a major concern is the use of heterogeneous tissues or mixed cells for methylation analysis, despite the fact that epigenetic marks are mainly cell type specific. For instance, the use of whole blood or placenta tissue in many studies is a significant limitation, considering that blood is comprised of various cell types. Methylation changes that are associated with BPA exposure can simply be caused by changes in blood cell composition as a result of exposure to not only BPA but also other chemicals and stressors. And a further concern is the use of potentially compromised study subjects. For example, women receiving reproductive medication or undergoing IVF treatment. The epigenetic changes that are reported in these women could simply be attributed to those therapeutic and not necessarily BPA exposure. So putting all this together, one can argue that drawing conclusion from the results of human studies that are published so far is very challenging, considering the quality of the available data and the design of those studies. So one option would be to consider future studies that are better designed and sufficiently powered and preferably done in a well-characterized population. Of course, this is not going to be an easy task considering the cost, time, and efforts that would be needed to carry out these studies and the wait time to get the results. The alternative and perhaps a more realistic approach would be to, if one wants to reach a faster conclusion, would be to focus on the available data from in vitro and in vivo studies. And this should be, of course, complemented with some follow-up studies, for example, using banked specimens that are already available from many of those published studies. For example, one can consider the functional consequences of the epigenetic changes that have been reported to be associated with BPA. This is a very important area that is unfortunately very understudied up until now. Along those lines, I think the report mentions the Clarity Project. Also, another source is the Target program that was funded by NIH several years ago. And one can maximize the use of banked specimens from those projects for preferably multi-omic studies. And doing so, we may contemplate integrative analysis of both epigenome and transcriptome in order to find the functional role of BPA associated epigenetic changes. And this can help us place this information in a wider context, which is the gene dysregulation and human disease, particularly human cancer. So I think I stop here.
Thanks, Dr. Besantinio. We'll move on to Dr. Wong with key characteristics 6 and 7. And again, I remind the discussants that we don't need a study-by-study description, but a summary of the key points that you think the committee should be aware of, particularly your assessment of the strength of the evidence of each of these characteristics.
Okay, can you hear me all right?
Yes.
So the first topic is chronic inflammation. So there was a handful of studies that were referenced in the report. I think this reflects that there isn't a whole lot of human studies that have been conducted on chronic inflammation and BPA. These studies, the report indicated that they reported an association. I would rephrase that and say that these studies suggest a link between inflammatory markers and exposure to BPA. And I think the major limitation in these human studies or epidemiologic studies is in the definition of what we consider chronic inflammation, which presumes long-term inflammation. And I think the major issue in defining this outcome is that the outcome assessment in many, if not all the studies, is made at a single time point, where any assumption of chronicity, in my opinion, cannot be made. So the way I interpret these studies that were referenced is that they have evaluated inflammatory markers, but not necessarily chronic inflammation. So this leads to the first major limitation of the delineated studies, which is study design. The majority are cross-sectional in nature, meaning that the exposure and outcome are measured simultaneously and at one time point. So we really cannot make any assumption about the chronicity of inflammation as an outcome, but we also cannot make any assumptions about causation. These are simply correlative studies. The second limitation of the reported studies is the consideration of covariates. Some, but not all, of the studies have accounted for relevant covariates, and the ones that have included covariates in their models, they all consider somewhat different covariates. And I'll just point out that there are actually a number of host characteristics and medical conditions that have been associated with inflammatory and other immune markers. And these include key cancer risk factors such as obesity, diabetes, NSAID use or other medication use, etc. Other characteristics such as physical activity have also been associated with immune marker measurements. So for some of the studies that have reported correlations but did not account for many of these covariates, the interpretation of the results is difficult. There was one thing that actually I found interesting about some of the studies that were presented, in that there were curious associations that, in the overall study, they perhaps didn't observe and associate on a link, significant odds ratio, but that many of the significant associations were actually reported among population subsets, such as women with PCOS or within diabetic individuals or among postmenopausal women. On the one hand, and likely these could just simply be spurious findings, but on the other hand, it made me wonder whether the question that was being asked by many of these studies is not quite the right one, that maybe we ought to be focusing on which susceptible populations there might be for BPA exposure, that there may in fact be a biological basis for concern for specific populations at risk. So if someone is obese or there's a diabetic population that already experiences chronic inflammation due to those conditions, might exposure to BPA exacerbate that level of inflammation. Now, concede that there's actually no evidence in the data presented to suggest that, but that's just another way of looking at it that we may consider in the future. The final limitation of the human studies is that power for many of these studies was uncertain. There was, for example, a cross-sectional study of 76 men in Italy, 200 adults in Korea, 176 healthy newborns in Cyprus, 40 women with PCOS compared to 20 controls in Italy, 60 adults in South India. There were some larger studies, upwards of 400 people, but still the population, as you can see, is very heterogeneous in terms of what we're looking at. And for the most part, the sample sizes are likely to be inadequate. Moving on to animal studies, there were many, many more animal studies on BPA and what I would consider chronic inflammation. And in these animal studies, longer-term BPA exposure was in fact measured, and chronic inflammation as measured within tissues and various organs, in fact, were reported. I think most noteworthy is that there were important dose-dependent associations with severity of inflammation increasing with BPA dose. There was a number of animal models that were reported. Long-term exposures were reported from over weeks to over months, and that were assessed. And there were even though these animal studies were also heterogeneous, because there were many more of them, and because they were covering many more exposures from weeks to months and from different doses and different routes of exposures, all coming up with similarly consistent associations, I found the animal studies to be a bit more robust than a lot more robust than the population epidemiologic studies. The biomarkers of inflammation were also measured in different ways, including in serum at different time points, mRNA expression at different time points, as well as directly in tissue based on inflammation, cell infiltration in the liver, kidney, lung, prostate, lung. So in general, I would say that the human evidence is weak to modest, but that the animal evidence of linking to chronic inflammation is much more robust. Moving on to immunosuppression, I don't want to spend a whole lot of time on this category because they're really very, compared to the other categories, there were very few studies to evaluate. The report suggests that there was that there is an association with immunosuppression. In my read of the data, I would say there's probably insufficient evidence. It's not null. Certainly there is the studies that they cited, there appear to be suggestion of an association with immunosuppression with BPA exposure. I guess where I had a little bit more difficulty in interpreting the results is that I don't know if it's a biological phenomenon, but unlike, we'll just contrast to the inflammatory studies, of which there were numerous, and they covered, many different types of exposures, many different types of outcomes over time. The immunosuppression studies, it's not clear whether these studies, because they're so few, whether they are spurious associations there, and they've blanketed sparsely different types of ways of measuring immunosuppression. So there's much fewer studies on dendritic cells or natural killer cells or IgM, and many more studies on macrophages and neutrophils and T and B cell proliferation. But even among those for which there's two ways to look at it. One is that because they're looking at all these different outcomes and there are studies popping up for each of these outcomes, that one could conclude that perhaps there is consistency in the association for immunosuppression. But the other way of looking at it is that there really wasn't any dose response relationship, but I don't know if I'm interpreting that correctly, that there's different dosing for BPA, and in some many of the reports will say there was a significant association for this level, but not at this higher level, or actually a number of studies do that. And so it's unclear whether there's no dose response or whether there is a threshold effect, right? So that maybe high BPA exposure, you tip the balance on immunity to actually inflammation rather than immunosuppression. So it's both sides of the coins, and none of these studies actually look at both sides of the coin. So I think because of the sparsity of the study on immunosuppression, it's difficult to interpret, would be my conclusion. And I'll end there.
Very good. Thank you, Dr. Wong, for a nice, concise, and informative summary. We'll go on. Key characteristic 8. Dr. Lamerrill, you have that one, receptor-mediated effects.
Hi. Sorry, I just had the sun come in. Okay, so receptor-mediated effects. I think that the evidence is overall strong here. There are a tremendous number of studies looking at bisphenol A's ability to bind and modulate the activity of estrogen receptors, both the canonical nuclear receptors of alpha and beta, but also the membrane receptors, and another cytosolic ER receptor called GPR or GPR30. And the studies have been done in cell lines of multiple species using gold standard techniques. And generally speaking, BPA appears to be an agonist of these receptors, like ER alpha and GPR. However, for beta, the ER beta, BPA can behave kind of as an antagonist. One of the things that happens with estrogen receptor is the effect of a ligand can depend on the tissue context or other contexts. For example, the chemotherapy tamoxifen can protect against breast cancer by antagonizing in the breast, but can promote uterine cancer due to differences in cofactors that are part of the binding complex of ER at the nucleus. And so, interestingly, estradiol itself also at ER beta kind of has antagonism properties. So that BPA can antagonize at ER beta is consistent with how the natural ligand estradiol works at that receptor. And ER expression in lymphoma cells that are human B cell lymphomas and characterized as ER alpha negative and ER beta positive, which is the predominant distribution of ER expression in B cell lymphoma patients and cell lines and so forth. That BPA exposure in these B cell lymphoma cells it did suppress the growth in the human cells and also in the mouse cells, which is consistent with basically how estradiol would work in that context, but not really consistent with what I described earlier with respect to the increase in lymphomas being reported in the animal studies. There's some evidence that BPA can be an anti-androgen in the human system. It seems to interfere with the translocation of AR and several independent studies of human cells in vitro. And thyroid hormone receptor has been evaluated. I didn't find that any compelling evidence related to BPA's ability to modulate the activity of thyroid hormone receptor. I didn't see any, for example, typical gold standard binding assays for that. The OEHA group was kind enough to summarize other receptors, including aerohydrocarbon receptor and 2 PPARs, alpha and gamma. I didn't find that there was consistent evidence of the the activity of either of those 3 receptors being modulated by bisphenol A. We also had a summary of the possibility that bisphenol A could modulate the expression levels of hormones or the receptors I've brought up. There could possibly be a role of BPA in increasing the expression of PXR, a receptor that was increased in association with BPA exposure in a study of infertile women and men, and also in fertile men, but not fertile women. So it's also been shown in female fish. Excuse me, it was absent in female fish, but also present in male fish, but not seen in non-human in vitro models otherwise. So I think that evidence is overall probably pretty weak. I would also say that there's inconsistent evidence for estradiol, progesterone, testosterone, thyroid hormones, thyroid receptor, and androgen receptor being associated with bisphenol A. In human studies, it's certainly been measured, but I think we've all discussed exposure assessment issues, but also I think outcome, there's a lot of temporality to the circulating levels of these hormones for people. So I think it's just would be very surprising if there was actually an association that represented biology that could be detected in the context of the experiments that we've seen. Just a little more evidence suggesting that there might be an effect on testosterone levels. I can get into that if someone wants, but my take on it was that it was not particularly important. Prolactin levels, the hormone, was positively associated with BPA in occupational studies of both sexes and in several rat studies, but non-occupational BPA studies, the prolactin evidence was inconsistent. So I thought that that could potentially be some evidence of something real going on, since the occupational studies address some of the issues that we've discussed in the previous conversations, and then with the rat studies supporting by having better control of confounding and exposure. Then just to touch on the AHR part of things, there was studies looking at several mouse tissues showing AHR protein was elevated, specifically male mouse testes, spleen, kidney, and those were 3 separate independent studies. And then in the human hepatocyte line, which is really a cancer cell line, had G2. And the AHR was measured as RNA in 3 cross-sectional studies in humans where it was also increased. I don't know what to think about that. I wouldn't really care about the cross-sectional so much on their own because of all the things that are wrong with that kind of study, but in combination with the cell and animal evidence, there might be something to that as well. And then lastly, I'll just touch on the PPAR gamma expression levels. There's increased PPAR gamma RNA, mostly identified, but just haven't looked at the protein as much. That RNA of PPAR gamma has been increased in experiments with human adipocytes, monocytes, and liver cells, as well as 3 different types of mice. And in those cases, it was mostly liver, and one of the 3 mice studies also had the testes data. And then there were a few mouse studies showing increased PPAR gamma at the protein level in a couple different tissues, liver and testes of mice. So overall, I would say that the evidence is strong, mostly based on the ER story, but I wanted to at least share with you all the receptors in case that's of interest related to some of the whole organism phenotypes we're discussing. Thanks.
Thank you. Let us move on now to key characteristic 9. And Dr. Landolf has that one, immortalization. Let's see, are you there, Dr. Landolf? Looks like you're still muted. If you need a minute to get ready, what we'll do is skip on to key characteristic 10, cell proliferation and nutrient supply. Dr. Bush has that one. So let's just skip forward. Dr. Bush, please.
Absolutely. Yeah. Thank you, Dr. Loomis. Okay, so key characteristic 10 alters cell proliferation, cell death, or nutrient supply. As the document states, there are over 200 relevant studies in human cells in vitro. Full disclosure, I did not read all 200. I did not get that granular. I did read the 3 comprehensive reviews and followed up as necessary. In general, I viewed the cancer cell line data of less value. As we know, transformed cells, and particularly these long established cell lines, already represent an artifactual system. So I'm also speaking as someone who uses these cell lines all the time in my own lab. So I gave more value to studies utilizing normal cells or non-cancer cell lines. As the HID document states, a number of these studies report no increase, and others report a decrease in cell proliferation. Of note, most of these studies used concentrations of BPA orders of magnitude more than what the physiologic concentration should be expected to be, in the range of 0.5 to about 4 nanomolar. And so there is going to be an effect, and there are many studies that report that BPA increases proliferation in various cell types, sometimes at a low dose, but this does indicate that BPA's effect may be impacted by cell type, the BPA concentration, and both extrinsic and intrinsic factors. In terms of apoptosis, many studies in human cancer cells demonstrating that BPA alters apoptosis signaling pathways and decreases apoptosis, but there are also negative results as well. Similar to cell cycle control and cellular replication, there is data to support a role there. There are a handful of studies that show BPA promotes angiogenesis and angiogenic pathways, and even the metabolic switch towards increased glycolysis. So I'm going to summarize quickly here. As part of this key characteristic 10, there was also evaluation of the in vivo rodent studies and the cumulative data there correlating BPA with proliferation in the context of hyperplasia. That was those conclusions ranged from some not statistically significant changes all the way to strongly supportive. So for KC10, in total, it seems that the way the evidence is presented, there is support for a moderate to strong consensus of key characteristic 10. And I will end there and let Dr. Landolf tell us about key characteristic 9.
Okay, thanks. Dr. Landolf is back on screen, so let's jump back to KC 9 immortalization.
Okay, let me get to it. So immortalization is a part of carcinogenesis. You can't have cancer if you don't have immortalized cells, because otherwise they'll just senesce or die out. So the data here is pretty thin. There are 2 studies that reported BPA could induce cell transformation in SHE cells, and 2 other studies in SHE cells, and one in A31, 113 clone of BALB-C3T3 cells did not find significant alterations to cell transformation, so they kind of cancel each other out. BPA did increase cell invasion in 3 human primary cell lines, and then increases in mesenchymal cell markers were observed in 3 human cancer cell lines and 1 human epithelial cell line after exposure to BPA, and 1 study observed a decrease in cellular senescence gene in the human cancer cell line. Higher urinary BPA levels in 1 study were associated with shorter relative telomere length in adult women. 5 studies characterized alterations to telomerase expression, activity, or telomere length after BPA exposure in human cells. 2 studies performed in primary human cells found decrease in telomerase activity, H-TERT mRNA expression, or telomere length after exposure to BPA. 3 studies performed in human cancer cell lines found increases or no alterations in telomerase expression and activity. So it's kind of a mixed bag. I'm not too wild about BALB-C3T3. I'm sorry, the SHE cell transformation assay. I know Carl Barrett and them have used it to study the temporal acquisition of transformed phenotypes, but I've talked to friends who have used this or tried to use it, and sometimes they get the same transformation frequency, but they score different events. So I'm a little bit skeptical about how reflective that is. I do know you do get immortalization after you get transformation of colonies, so it can do that, but you kind of got these studies canceling them out. So I would say the more interesting studies are probably in telomerase activity area, but this database is pretty thin. So I would say, yeah, it can cause cell transformation, but the studies, in some studies, but they're canceled out by other studies. I think the most interesting data is the studies in the primary human cells where the telomerase activity is decreasing and H-TERT expression or telomere length after exposure to BPA. And 3 studies performed in human cancer cell lines found increases or no alterations in telomerase activity and expression and activity. So I think that database is kind of mixed. And I don't think it conclusively shows immortalization. Some studies being positive, some studies being negative. So I would say there's not a lot of data here at all for causing immortalization, in my opinion.
All right, thank you, Dr. Landolf. So that brings us to the end of the discussion of the key characteristics. We have some time set for committee discussion right now, and the way I'd like to do this is first to call on the other discussants who reviewed and reported on the animal and mechanistic data and see if there is any additional information that they'd like to bring forward or any perspectives that we haven't heard yet on the data that have been summarized by the other discussions. So Dr. Bush, hand up there, go ahead.
Thank you, Dr. Lewis. So by my count, for the key characteristics, I got about 6 or 7 of these key characteristics that I think folks were generally would say had strong associations, and maybe key characteristic 3, 7, and 9 would be of limited, and I want us to be careful there. development of the key characteristics is certainly a great utility to the community. It's definitely valued and provides that framework, that agnostic framework to build upon when it comes to carcinogens. But there's also emerging data, indicating that there needs to be some revision of these key characteristics. And I'm going to point to a 2019 review by Krusky and others. In fact, our own Vince Cogliano was on that paper, where they looked at the 86 agents known to cause cancer in humans. And so we're looking at the group 1 agents, and on average, group 1 agents have about 4 of these key characteristics. None have 10, only about 20 have 6 or 7, as we've indicated for BPA here. And those group 1 agents include asbestos and plutonium and tobacco. So I say this to perhaps help guide the conversation in terms of what these key characteristics represent. So with a little bit of caution.
Thanks. All the points are well taken. I'll go on to Dr. Lamero.
Yeah, I just wanted to ask a question. Hopefully, Dr. Stern, you could address it the best. There's several meta-analyses of adults and kids, as well as actually experimental rodents, indicating that BPA is associated with increased risk of adiposity. And of course, in rodents, it's experimental. So I was wondering if we're conceiving of BMI as a mediator rather than a confounder, how might that change your perspective on some of the epidemiology studies you reviewed? I think breast cancer certainly is associated with obesity, and then prostate and gallbladder as well.
Yeah, that's a great point. And there are, and I can look through my summary and point those out, but there were a few studies, only a handful, that actually explore that possibility, and they look for potential effect modification of BMI as a surrogate for obesity on the association between BPA and cancer risk. And actually, they did not see evidence that BMI was an effect modifier. The other studies either did not consider BMI at all, and that was a problem, particularly for breast cancer, because as we know, obesity is a risk factor for breast cancer. And some of the studies did consider it as a confounder, so they adjusted for it, but they may not consider the potential effect modification role. So only a few studies look at that, and if I remember correctly, I want to double check now, they did not find evidence that there were different associations by BMI status. But only a handful of studies looked at that. Majority did not consider it.
Mm-hmm. And no one did mediation analysis. So
no, no, but by doing the effect modification, we can kind of infer at a potential mediation. If we did see, for example, that among women with higher BMI, the effect of BPA is stronger for like breast cancer risk, then we can kind of speculate that, further analysis could look at that, but only a few studies look at that. So, and there was no proper mediation analysis done in any of the studies.
Okay, thank you.
Okay, Dr. Wong has a hand up as well. Go ahead, please.
Yeah, I just wanted to reiterate, actually, I wanted to jump onto what Dr. Bush had along the same line. For some of the epidemiologic evidence or the human studies that I alluded to for my section on chronic inflammation, it's not that there were, and I think Dr. Mack touched on this previously as well, it's not that there were no studies necessarily, it's that the study designs or the studies are not adequate. They're not, they're not designed in a way to assess exposure adequately or their outcomes adequately. So I think that's what we have to consider. It's not that there are, a slew of null associations that we're working with. And I did have one question for Dr. Stern. I didn't see, and I didn't hear in the summary, but I thought I saw in the literature. Can you confirm, were there no studies on children or like in utero exposure and then childhood cancers?
No, there were no studies,
So-
either of those. All studies
Okay.
were on adult populations.
There was one study of osteosarcoma,
Oh, that's true, with
which most
younger,
of them were
yeah.
pediatric patients.
Yeah, that is true. The study in osteosarcoma had adolescents, I think, yeah. But no studies of exposure in utero.
Thank you.
Okay, Dr. Eastman, another comment?
Yeah, I just thought I'd expand a little bit on a question I asked earlier, but it's more of my perspective. But when you have lots of multiple studies and indeed the Clarity study has 7 different arms, and all of these have many, many types of statistical analysis, literally hundreds and hundreds. What I start looking for is, rather than statistical significance on any one test, but looking at sort of, do we see reproducibility and consistency? And I think that's really important. And in some cases, I think it's important to go back to the original study. And that's why I referred to the NTP 1982. If you go back and read what they commented about specific tumor types and their interpretation, and the same thing with the Clarity, and reading also the FDA letter that was submitted recently in the public comments, talks about some of the issues related to statistical analysis and how this was done and combining and their interpretation. For me, I think it's this is helpful to look at and looking for consistency. And I haven't seen it yet, but maybe others see that. Anyway.
Okay, thanks. So that's an important question, but the way I'd like to organize the discussion here is just that we have a public comment period coming up and then another committee discussion period after that. And I'd like to hold discussion that's leading to a listing decision for that next committee discussion session. So if there's a different question from members of the committee, let's take that now, but kind of focus on the evidence that's been presented already rather than the listing decision. So Dr. Lamerrill, does your comment fit into that category?
Yeah, Dave Eastman asked about consistency, and I wanted to remind him that in the NTP 1982 study, the male B6C3F1 mouse had increased malignant lymphomas, as did, of course, the Clarity study. And so I agree with his idea to look at consistency. It's a nice criteria to contribute, but we do have 2 independent studies in 2 species with a malignant lymphoma. So I just want to clarify that.
Okay, thanks. Important point.
Isn't
Dr.
that the
Warren?
rat, by the way?
I'm sorry, I didn't hear the question.
I thought the one in the 1982 study was in the rat.
In
but-
my notes, it says B6C3F1 mice, but I can double check the.
Okay, I'll look too.
Okay, Dr. Wong.
So I hope it belongs here, but I do share some of the reservations that Dr. Eastman has expressed in reading. I appreciated the statistical overview at the beginning of the presentation, but in my mind, it would be helpful to see a rebuttal of many of the points that were made in the FDA letter. I don't know if that's appropriate here, but.
Now let's take that one up after the public comments. Okay, Dr. Romero, you still have your hand up. You have something else that you wanted to bring up?
No, I apologize. I'll use the chat to put in the point.
Okay. Okay, good. Any other questions about the evidence or additional points that the committee would like to bring forward right now? Okay, so the next agenda item is public comments. And we probably should do a survey and see how many comment cards we've got before we start.
Hi, Dr. Loomis. We've received 6 comment cards.
Okay. So
Dr. Loomis, before going in, I think Vince Cogliano had his hand up and wanted to just clarify one point that was
Okay.
raised in the committee discussion.
Sorry, I didn't
Before
see
we move
that.
to
Go ahead.
comments. Yeah.
Yeah.
Vince?
Thank you. I had my hand up just for a little bit because I think you got it, but the key characteristics paper that I was a co-author on, there were quite a few results in that, and I think the idea is that really we didn't find any of the group 1 carcinogens that had all 10 key characteristics, and the average was about, as Dr. Bush said, around 4 or so, and there were somewhat more, but there were also somewhat less. So, but we don't really look at it as how many key characteristics. It's the key characteristics tell us how an agent might cause cancer. What are some of the major pathways or events that are going to be involved? But definitely we don't need all 10. That's all. Thank you.
Okay, thank you. So there's nothing else before public comments. So we have 6 cards. Speakers are limited to 5 minutes each. That's about 30 minutes, more or less. So I'm going to suggest that we go ahead with public comment and then call a break after that and return to committee discussion, if that sounds okay to the staff and committee members. Any objections? Okay. So Elizabeth has put up the procedure for public comments. Just really briefly, one has to be registered for the Zoom webinar in order to make a comment orally. You can do this in 2 ways. You can fill out a speaker card. You can see the URL for that here on the form. Or you can raise your hand in Zoom, and then when you're prompted to speak, unmute yourself. And remember to lower your hand again when you finish speaking. So the staff will keep time. I'm not sure who's doing that, but they'll let you know when the end of your 5 minutes is approaching, if you haven't finished. And at the end of 5 minutes, the microphone will be cut and we'll move on to the next speaker if you haven't finished. So I don't have access to who has submitted a speaker card. So I think Dr. Gilson will go ahead and call on the speakers in the order in which they registered.
That's right. Thanks, Dr. Loomis. So first up, we have Robin Pruitt. If you're here, can you, I'll go ahead and unmute you. And if you can put up your hand, it'll make it a little easier for me to find you and allow you to talk. There we go. You should be able to go ahead.
Okay, great. Can you hear me?
Sure can.
Great. Well, thank you for the opportunity to speak today. My name is Robin Pruitt, and I'm a board certified toxicologist at the environmental consulting firm Gradient. And today I'm speaking on behalf of the American Chemistry Council. So the OEHA guidance criteria for identifying chemicals for listing as known to the state to cause cancer indicate that if the weight of the evidence clearly shows that a chemical causes invasive cancer in either humans or animals, then that chemical may be listed. Upon review of the body of evidence included in the OEHA hazard identification document for BPA and considering the strengths and limitations of the reviewed studies, BPA does not meet the criteria for listing. As discussed in more detail in the written comments submitted by ACC, the epidemiology studies of BPA and cancer have multiple limitations that can bias the results in either direction and not solely toward the null as indicated in the hazard identification document. Despite the limitations of the epidemiology studies, the results across analyses of specific cancer types are inconsistent and do not clearly show that BPA causes invasive cancer in humans. The evidence from experimental animals also does not clearly show that BPA causes invasive cancer. The majority of the animal studies reviewed in the hazard identification document have significant limitations, such as short study durations and small numbers of animals per dose group, and these studies are of limited utility for assessing BPA carcinogenicity. In contrast, the chronic rodent carcinogenicity study conducted by NTP in 1982 and the Clarity BPA core study are well-conducted, high-quality studies that evaluated BPA over a wide range of doses. The NTP study reported increased incidences of a few types of tumors in male animals, but these do not provide strong evidence for BPA carcinogenicity. Consistent with the conclusions of the study authors, it is more likely that the cancer types observed in this study were chance findings or common spontaneous tumor types in aging male rats. The hazard identification document fails to mention the statistically significant decrease in adrenal tumors in rats, which suggests that the large number of tissues and endpoints examined in this study, as well as the multiple statistical tests used, may have led to false positive or negative findings that are due to a lack of adjustment for multiple comparisons rather than to BPA exposure. The Clarity BPA study reported very few statistically significant increases in the incidence of malignant tumors in rats that are likely the result of chance fluctuations in incidence or false positive findings due to lack of adjustment for multiple comparisons. Despite the reliable evidence from this study that is consistent with the lack of BPA carcinogenicity in rats, the hazard identification document presents a biased review of this study that appears to be aimed toward a conclusion of carcinogenicity for BPA. This was also noted in the written comments submitted by the FDA. For example, the OEHA authors conducted their own statistical analyses of the Clarity study data without any rationale for doing so, and this practice can lead to the reporting of false positive results. The OEHA authors also used unreliable historical control data sets to attempt to identify rare tumors, but this analysis is inappropriate and goes against the evidence that these tumors are spontaneous and not statistically increased compared to the concurrent controls. The hazard identification document also discusses several issues that have been brought up in the literature regarding the Clarity study, such as background contamination with BPA and controls, potential insensitivity of the rat strain to known estrogenic chemicals, and the lack of an unhandled control group. However, the hazard identification document did not mention that all these issues have been addressed by the Clarity BPA study authors and have been shown to not limit the ability of the study to detect carcinogenic effects. Consistent with this, the FDA made the following statements in the comments that they submitted to OEHA. I quote, "Numerous errors and incorrect or inappropriate analyses of the Clarity BPA core study results have been identified. We recommend OEHA consider these issues for reanalyses as the current methods applied by OEHA lead to an unsupported conclusion of potential positive carcinogenicity of BPA. FDA's multiple evaluations examining carcinogenicity and the results of the Clarity BPA study do not support classifying BPA as a carcinogen." With regard to mechanistic evidence, the hazard identification document focused on providing any
1 minute left.
Any positive evidence for each of the 10 key characteristics, but did not weigh the evidence for or against each as a plausible mechanism for BPA carcinogenesis. The 10 characteristics are also shared by many non-carcinogenic substances, so the existence of evidence for these characteristics for BPA in certain studies does not provide strong evidence that BPA is carcinogenic. So overall, the available studies of BPA do not provide clear or consistent evidence that BPA causes invasive cancer in humans or animals. Therefore, they do not meet the OEHA criteria for listing BPA as a carcinogen, and BPA should not be listed as such. Thank you.
Thank you very much. Dr. Loomis, back to you, or we can take the next comment.
Yeah, just go ahead. I don't know who the speakers are, so if you would just go ahead with the next one when each finishes, that would be perfect.
Okay, wonderful. So next up we have Katie Pelch, and I'll go ahead and prompt you to unmute. Are you able to unmute yourself? Oh, I see it. Go ahead when you're ready. You should have permission to talk, but I'm not hearing you. Okay, we'll try to come back to you.
Yeah, let's go to the next speaker.
So the next speaker is Rainbow Rubin, and Rainbow did have to leave. So they provided a public comment that I'll read on their behalf. So this is from Rainbow Rubin, PhD and MPH, Director of Science with Breast Cancer Prevention Partners. Evidence from animal and in vitro studies has suggested an association between increased incidence of breast cancer and BPA exposure at doses below the safe reference doses that are the most environmentally relevant. BPA may increase mammary tumorigenesis through at least two mechanisms: molecular alteration of fetal glands without associated morphological changes, and direct promotion of estrogen-dependent tumor cell growth. Both results indicate that exposure to BPA during various biological states increases the risk of developing mammary cancer in mice. In human breast cancer cases, urinary BPA concentrations have been positively correlated with breast adipose tissue BPA in the case group. BPA mimics both the structure and the function of the hormone estrogen and disrupts endocrine function at very low doses, consistent with its non-monotonic dose response curve. There is no evidence to suggest that reverse causation is the mechanism of action in breast cancer, as suggested by a committee member. Just because no study accurately captures BPA exposure during vulnerable stages of development doesn't mean BPA is not a concern. Just because study results are inconclusive does not preclude us from protecting the population from BPA based on the precautionary principle, considering the structural and functional overlap between BPA and estrogen. Establishing a causal relationship in humans is extremely difficult due to multiple and variable exposures. As Dr. Max said, there are no evidence against a positive association. Today, we heard ample evidence that BPA is a carcinogen, mutagen, and reproductive toxin. Please move to add BPA to the Prop 65 list. All right. Now I see Katie's hand is up again, so let's try again here. Please go ahead. So we hear the sound is breaking up a bit. Try again.
Okay, can you hear me?
Yes.
Okay, thank you. I am Dr. Catherine Pelch. Thank you for having me here today and for allowing me to provide comments. I am a scientist at Defense Council. We're specialized in evaluating the hazards and risks associated with exposure to chemical BPA. To the staff
excuse me,
for
if I can
their
cut in here. The sound quality is pretty sketchy.
Okay, can I try to call?
Go ahead, yeah.
Okay, I will hang up and I'll call back in a moment.
Okay, great. Thank you.
Maybe we can go on to the next speaker then.
Very good. So the next speaker is Luisa Camacho. And if you can raise your hand, that'll facilitate my unmuting you or allowing you to unmute yourself. All right, I'm not seeing them here. This was Luisa Camacho with the FDA. So let's go on again. So the last two speaker card submissions were from Anon, so anonymous. If you're here and you would like to provide oral comment, please go ahead and raise your hand, and I will allow you to unmute yourself. All right, seeing none, I'll just summarize what Anon provided in the public comment card. So there are two submissions. The first one says, presentations have ignored public comments submitted to the OEHA document. And then the second comment, also from Anon, is that public comments appear to be hidden from consideration. And then they provide the website where public comments are posted. So to hopefully address these points, the speaker request card is to indicate if you would like to provide an oral comment during the hearing. So written public comments regarding the hazard identification document that were submitted during the public comment period are for the CIC and were provided to the CIC as part of the hazard identification materials before them today. And these comments are publicly available on our website. Now, until Katie Peltz calls back in, that rounds out the public comment cards that were submitted.
Okay, I guess we can see if anyone else has their hand up to make a comment who hasn't submitted a card yet.
So if you'd like to provide public comment, but didn't submit a speaker request card, please raise your hand in Zoom at this time. All right. No one has their hand up.
Okay, and Katie Pelch hasn't called back in yet, I guess.
Let's see, I have a hand up here from Katie. Sarah, if we can try again.
Okay, let's try.
Okay. So you should now be able to unmute yourself and provide your comment.
Can you hear me now?
Yes.
Okay, perfect. Thank you so much. Sorry about all the technical difficulties. I am Dr. Catherine Pelch. I am a scientist at the Natural Resources Defense Council, where my expertise is in providing hazard assessment and risk assessment for chemicals like BPA. So I just wanted to start off by thanking the OEHA staff and the CIC members for undertaking this, this very Herculean effort to evaluate the evidence for BPA, and want to encourage the CIC to consider the guidance that says that the mechanism of BPA, or understanding the mechanism for how BPA may be a carcinogen, is not necessary. Rather, it is important to look at the entire body of the evidence. And I think that we have heard today through OEHA's immense efforts to catalog the data that there are numerous studies that show that BPA has the potential to be a carcinogen, both the animal evidence and the mechanistic evidence. And we also heard that it is not necessary for BPA to meet all 10 key carcinogen or key characteristics of a carcinogen, but we did hear strong evidence that BPA is meeting several, perhaps 6 or 7 of these key characteristics. So I just wanted to quickly echo the comments that I submitted in written form. I apologize as I'm driving right now, so hopefully you are able to hear me, and want to encourage the CIC to list BPA as a carcinogen.
Thank you.
Okay, very good. Let's do a final check and see if there are any other hands up for public comment. Doesn't look like it, right?
Right.
Okay. Well, it's a good idea to take a break sometime, and we're past the time program for that on the agenda. So if there's no objection, I'm going to suggest we take 15 minutes right now and reconvene promptly at 3:30. Okay. So I'm not hearing any objection. So the Bagley-Keene warning still applies. Carol's not here, but I think we all heard it. Anyway, 15-minute break.